Electroporation is a technique where an electrical current is applied to live cells in order to open transient pores on the cell membrane.
Through these pores, dyes and other molecules that normally do not cross the cell membrane can enter the cell. The pores reclose quickly, with no damage to the cell.
We will describe how adherent cells can be electroporated as they are growing on a slide that is coated in part with electrically conductive and transparent material. The material is indium-tin oxide, and as it conducts electrical current, it opens pores that allow a dye to enter the cell. Then it is possible to examine if the cells have gap junctions or not, and how many. Gap junctions are small channels that connect the interiors of neighboring cells and allow the passage of small molecules. It has long been recognized that gap junctional communication has a variety of important functions in the cell, including neoplastic transformation and metastasis.
Welcome to this Cell Projects presentation about how to perform adherent cell electroporation using the in situ porator. This image shows what you will be able to do. The fluorescent dye, lucifer yellow, was introduced into cells growing on a transparent and conductive glass, up to this line. The line is easier to see under phase contrast. Here it is. You can see that the dye has diffused through gap junctions from the cells on the left, which have been loaded with dye by electroporation, to the cells on the right, resulting in this gradient of fluorescence. Cells are trypsinized, then transferred into a tube and centrifuged.